This video will be looking at eukaryotic control mechanisms for gene expression. Gene expression is a simple concept. It is the idea that a gene is being expressed, meaning that a protein is being made of some kind. Eukaryotic organisms do control the expression of genes. Not all proteins are being made all at once, and so in order to control gene expression, there are certain things that occur. Just a quick review, the difference between chromatin and chromosomes. Chromatin is found during the interphase part of the cell cycle, and chromosomes are only present during cell division. Now, during the chromatin phase, the DNA, or portions of the DNA, are wrapped up on structures called histones. And a histone is just a small little protein that acts like a little spool. You can see here that the DNA wraps around each histone a couple times. And normally during the chromatin phase, these histones are pretty loose, but the DNA is still bound up on them. You can see here in this picture how when the chromosomes are being formed, those histones come real close together, form these units, and get real big. That's supercoiling. But most of the time, the DNA is just found kind of like this. Well, when a DNA is on the histone, that DNA is not able to be transcribed and then translated, obviously, because the histone's in the way. We call that DNA that is condensed, that is not active, that is currently wrapped up on the chromosome or on the histones, we call that heterochromatin. So these are genes that are unable to be expressed because they are currently on histones. The genes that are active, that are able to be expressed, are called euchromatin. And so if you can look in this picture, you can see the dark spots are all the parts where the DNA is still wrapped up, and the other spots are where the DNA is actively being transcribed. Typically that active transcription is happening in this region called the nucleolus. And so we're going to look at some of the different ways that can occur. Most of gene control happens to prevent transcription and at that stage. There are some that transcription occurs, and then there are some other stops that are going to take place to look at, but a lot of them happen prior to transcription. One of those is called DNA methylation. DNA methylation is the idea of putting a heavy coat of methyl on the DNA, and methyl is just a CH3. We've dealt with these groups before, methyl group, and you can kind of see here that when that methyl group is added to the DNA, what this does is it causes it to not be able to be transcribed. It basically locks up that section of DNA. There is some other things that can happen as a result of DNA methylation. One of those is called genetic imprinting, and this is basically where sections of the DNA have been methylated, and even though you may have those genes are unable to be expressed. And so maybe you have a section of your genome that has been passed down to you, but it's been passed down to you as methylated genes, and those genes are unable to be expressed, and that can actually affect your appearance or other sorts of things that are body processes that are going on. Another thing that can affect or control transcription is called histoneacetylation. And this is where these groups called acetyl groups, we've talked about acetyl before, this is what acetyl looks like, or not that important, other than to say acetyl causes the bond between the histone and the DNA to no longer exist. The histones have a positive charge, DNA has a negative charge, and so they really like each other. Well, the acetyl group takes that positive charge off the histones, propelling the DNA. And once this happens, of course, then DNA can be transcribed. And so this is a little bit opposite of the methylation, because this allows for transcription to occur. Some of these happen, some of these pre-transcription factors or during transcription factors actually can occur during the building of the factory. And let me explain how that works. And so here's the transcription initiation complex and RNA polymerase. And in this particular picture, there are these structures called enhancers. And what the enhancers will do is they will actually kind of bend over. As you can see, this bending protein here, not important, but it does cause the DNA to bend over, and essentially what's happening is it is pushing that transcription initiation complex along the gene. If you have a wagon and the wagon is slowly rolling downhill, well, that's fine, but you can make it go faster than it is by pushing it. And so this enhancer causes that to occur quicker. In the same way, there can be repressors that stop that from happening. I believe I have a picture of the repressor. Here's just another picture of those enhancers. Repressors can stop that transcription from occurring. After transcription has occurred, there are tiny segments of RNA called microRNA and small interfering RNA, miRNA, and siRNA. And what these do is they actually attach to portions of the transcript of the messenger RNA and prevent it from being translated. And so you could think of this as a pre-translation kind of blocking, because after it has already been made, this piece of RNA keeps the ribosome from binding or prevents the ribosome from translating that all the way. Another type of control is after the transcript has been made, the spliceosomes, remember they take out the introns and they leave in the exons. Well, that can actually change that gene. If you mix, or take these two, if you mix exon 1 and 2, if you swap them, the protein is going to be different. And so we've talked about this. We called it alternative RNA splicing. That is another type of control mechanism that can occur. There are several others that we could talk about. After translation, the protein folds up, and that protein folding can be changed, manipulated, and that sort of thing. But the main thing I want us to talk about is just how that is controlled and how the product can be controlled. And so we've covered a list of ways that can occur.